Hello students, today we are going to learn a new concept, that is Human Genome Project, which is coming under your chapter 6, Molecular Basis of Inheritance. Under this concept we are going to explore introduction, goals of Human Genome Project, its methodology, features of human genome and its application. What is genome? Remember, genome is the entire genetic material present in an organism. No two individuals are similar because they differ in genetic makeup. Difference in genetic makeup is due to difference in nucleotide sequence. Remember, difference in nucleotide sequence present on the DNA. So, it was an ambition of scientists for a long time to map the entire human genome. Advances in genetic engineering such as isolation of DNA, cutting the DNA, then cloning of DNA and sequence the base pair, nucleotide base pair present on the DNA fragment led to this happen. In 1990, US Department of Energy with National Institute of Health had started a mega project which is popularly known as Human Genome Project or HGP. Wellcome Trust of UK also joined in this project as a major partner. Later, China, Japan, France, Germany also joined in this mega project. Human Genome Project is known as a mega project because it needed a lot of money, advanced technology, advanced computer and scientists. For an example, to sequence a single base pair, it is $3 American is needed. For a single base pair, $3 American is needed. In human genome, there is 3 into 10 to the power 9, that is more than 3 billion base pair are there. So billions of dollars is needed in this project. Got it? For storage of the sequence data, if one book has 1000 pages, then 3300 of books is needed to sequence the genome of a single human cell. So, an advanced computational device is needed for store the data and analysis it. Okay, this lead to open a new branch of biology which is popularly known as bioinformatics. When information technology is associated with biology. Okay, so that we store a lot of data and analysis it. Got it? Then come to goals of Human Genome Project. Goals of Human Genome Project HGP. To determine the sequence and number of all base pair present in human genome. Determine, the first goal is determine the sequence and number of all base pair present in human genome. Then identify all genes present in human genome. Let's come to next. Then determine the function of all genes. Determine the functions of all genes. Got it? Determine the functions of all genes. Then store the information in database. Store the information in database. Store the information in database. Improve tools for data analysis. Improve tools for data analysis. Okay. Then transfer of technology developed during HGP or Human Genome Project to industry. Transfer of technology developed during Human Genome Project to industry. Then ethical, legal and social issues, popularly known as ELSI, must be addressed and solved. This mega project took 13 years. That is from 1990 to 2003. Got it? Next, come to its methodology. In methodology, there are two types of approach for analyzing of genome. First one is identify all genes that expressed as RNA. This technique is known as expressed sequence tags or ESTs. Got it? The second one is sequencing the whole genome. Which is popularly known as sequence annotation, popularly known as sequence annotation. Human Genome Project adopted the second methodology for genome mapping, that is sequence annotation. In this methodology, the points are: first, full DNA of cell isolated and broken into fragments. Whole DNA of cell isolated and broken into fragments. Then the fragments are inserted into specialized vector like bacterial artificial chromosome, yak yeast artificial chromosome for cloning. Remember, PCR, polymerase chain reaction, are also used for cloning of this fragment. Okay, then fragments are sequenced as annotated DNA sequence. Then fragments are sequenced as annotated DNA sequence. This methodology was developed by a famous scientist, Friedrich Sanger. The annotated DNA sequence, this methodology was developed by a famous scientist, Friedrich Sanger. Then the sequence were then arranged on the basis of some overlapping region. Okay, the sequence were then arranged on the basis of some overlapping region. It generates the overlapping fragments, which generates overlapping fragments. It generates overlapping fragments for further sequencing. Got it? In next step, computer-based programs were used to used to align the sequence. Align the sequences. Then computer based program were used to align the sequence. Then the sequence were then annotated. The sequence were then annotated. Sequence were then annotated and assigned into different chromosome. Remember, all chromosome of human, that is 22 pairs of autosome, X chromosome and Y chromosome sequenced already. Chromosome 1, remember, chromosome 1 last to be sequenced on May 2006. Got it? Then comes to its features of human genome. Features of human genome. Features of human genome. Features of human genome. The human genome consists of 3.1647 billion nucleotide base pair. An average size of gene contains 3000 nucleotide base pair. Average gene contains 3000 nucleotide base pair. The largest gene is Duchenne. Largest gene Duchenne muscular dystrophy present on X chromosome. Present on X chromosome, having 2400 kilobase pair. 2400 kilobase pair of nucleotides, and the smallest one is beta globulin, and insulin gene, which has up to 10 kilobase pair of nucleotide. Got it? Then there are more than 30,000 genes. There are more than 30,000 genes present in human genome. There are more than 30,000 genes present in human genome. Chromosome 1 has most number of genes, that is your 2968 gene. 2968 genes present in chromosome 1. In Y chromosome there is 231 number of gene, and in chromosome 1 2968 number of genes are present. Okay, function of 50% of the gene, discovered gene. Function of 50% of discovered gene. Function of 50% of discovered gene is still unknown. Only less than 2% gene, less than 2% of total genome coded for protein, which is known as structural gene. Which are known as structural gene. Got it? Then 99.9% of total nucleotide sequence, 99.9% of total nucleotide sequence are exactly similar in all humans. Only 0.1% of total genome, that is your 3 million base pairs, are responsible for the variety occurs in human beings. Got it? 1.4 million location at about 1.4 million location, there is SNPs, that is single nucleotide polymorphism. Occurs, that is single nucleotide polymorphism occurs. It has potential to trace the human history. Okay, we'll study it in further concept, that is your DNA fingerprinting. Okay, then come to repetitive sequence. There are two types. One is your mini satellite and another one is your microsatellite. There are 30,000 mini satellites are present. Okay, mini satellites are range between 11 to 60 base pair in long and repeat tandemly up to 100 to 1000 times. Tandemly repeat. Tandemly means it repeats to its adjacent site. There are more than 2 lakhs. Remember, there are more than 2 lakhs of microsatellites are present, and its length is about 1 to 10 base pair, and the tandemly repetition and the repetition is up to 10 to 100 times. Okay, then comes to the application of Human Genome Project. Application. Human Genome Project has a large scale of application. They are applied in applied biology. A section is there that is applied biology. Okay. Human Genome Project poses very important role in finding the human history or tracing the human history, human evolution. Got it? Remember, more than 1200 genes are responsible for the common disease, your cardiovascular disease, endocrine disease like your diabetes and neural disorder. Okay, and most important is your cancer. Okay, in healthcare system, using human genome project, using the data of human genome project, we have developed many vaccine which have positive impact on human body. Okay, then the interaction of gene and protein which is interconnected to each other is well studied in applying the data of human genome project. Okay, the transcript means the RNAs. Okay, the transcripts and the genes from tumor tissue are analyzed and known its effects. Okay, analysis of these with the data that is sequenced in human genome project. So we know that human genome project has a large scale of application.